Hello class, good evening. Good evening बच्चों, क्या हाल है? How are you all? So you all will be from the diverse part of India, everywhere from north, south, west, east. That's the best part, like meeting everyone uh, and joining you all together. Um, that's beauty of these workshops. Otherwise, if in the class we are teaching of one region people only, but these online workshops are bring everyone together, diversity in unity. So I would say, oh, unity in diversity. Sorry. Anyhow, so are you ready for today's lectures? So today we will be dealing with somaclonal variations and somatic embryogenesis, synthetic seeds. That three topics will be covered mainly, and so we will understand their applications, their methods, how they are done, gametoclonal variations. All these will be covered today. So who wake up today at 5 a.m. to go for exercise, like we discussed yesterday, or waking up at around 7, 8 than normal times? No one, no one. Okay. Good evening, Bachchu. Good evening. Somebody wrote me that they want to send me handwritten assignments. Yes, yes, you can send me handwritten also. I'm fine with it. At the end of the day, it's gonna be with you, so you will be in charge of your own assignment, so that you will remember that what you have done on your own. So that's the main idea behind the assignments. And some of the students from our MBBT, they have already published in some journals, free journals, and like five students they get together and they went to the journal, and fact paper was around 5.45, and it's done, voila. So all is done, so their review paper is accepted, and it's all done. So there are some students who are already doing without any support or something. um, so I will also share those links here. Those students who have did it. I share on your um, Telegram group. So have a look. It's not so good general, but still to start your career, somebody's in bachelor studies. If you can publish it, most of those students were on the bachelor studies actually. They who published it, so it will be really nice from the reviews if you can publish something out of it. Mm. And yeah, so it's 8:02. We have very few students joined us today, not so many. But we can, shall we start then? Or in meantime, I was thinking, uh, How about what if we are having these normal lectures? I also touch with you some of my work that I did, which you could use in your research point of view. So maybe I can teach you how Western blots, how mRNA, how microscopy, all these techniques could be used in plant biotechnology also in your research area. You can compensate those techniques and how you um, analyze the data. How do you interpret your data? Then data interpretation is also very important. How do you plan your data, right? How do you plan your experiments? How do you take care of your controls? How do you take care of your positive controls? How do you take care of negative controls? So I was thinking also to add to our part. So maybe we start with that then. Yeah, a little bit about ER stress signaling that's also happening in the plant biology, because at the end of the day signaling mechanisms, they are all same in human and also in the plants. It's just there's little bit change up and down. Let me just confirm it whether the ER signaling ER stress signaling बच्चे it's ER stress signaling, it's related to the ER endoplasmic reticulum, plant immunity. Yeah, okay, now uh, so there's so they also have the same molecules as we can see. And there's a PERK, RE1 alpha, S1MLT, S1PS2P, Golgi body. So there's not so different. So what we're gonna study is also same in the plant. So don't worry. Right. So let's start the journey. Mhm. Just a second. Let me set up my pen with it, and also. There's a lot of background noise. Um, no. So, let's start this lecture of ER stress signaling. So, I will be dealing with you today one of my thesis work in my PhD, my second project. Mainly, the reason to show you this project is to help you out how to understand a project, how to create your hypothesis, how to how to plan your experiments, and how to um, yeah, arrange things for the later. So this is will give you a bit summary. So I didn't choose my first project because that will be too much for you to understand. But this is smaller project and it's somehow correlated with uh, plant biology also. So in normal cases, so there are some proteins called as folding proteins. So in our case here I choose CTGF. connective tissue growth factor, which is present in the ER lumen. And beyond that, there is a ER stress, as we can see here. So this ER stress might sometime could make stressed our ER lumen. And in the ER lumens, we have PDIs proteins, yeah, we have PERK proteins, we have RE1 alpha proteins, we have ATF6 proteins, and all of them, they are being attached with the down here the BIP. That's also kind of a messenger to it, being attached to all the proteins and letting them in their inactive state. So they are not in active state at the moment. So what once there is a dimerization is happening here by the phosphorylation also. So two perks comes together, two perks come together, and what they do the EF2 alpha phosphorylation. They do a kinase activity by phosphorylating the EF2 alpha, and this EF2 alpha will further activate ATF4, or it will stop the translation. It will inhibit the translation, and this ATF4 protein, with the help of CHOP proteins, it will do the transcription, folding of enzymes, and upregulation of enzymes also. So so that's the one signaling pathway of PERK alpha, like till here, from here till here, and here, 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 and here till there. So then RE1 alpha, so it's also got phosphorylated, it got dimerized, but what it does afterwards, it's make XBP1, another mRNA, to be spliced. So it makes make it spliced version of that, and then that one is translated, and then after translation, it's also do the same things. On the other hand, ATF6 alpha, it's already being then cutted with S1P, S2P. These proteins are connected to ATF6. They got attached to the Golgi body and they are cleaved. And you have this cleaved ATF6 then, upper version. They are then do the same functions. So these are the various markers of ER stress. If you see any increase in the splicing of XBP1, or increase in ATF6, ATF4, or you see ATF6 to be cleaved version in the nucleus, being cleaved going in the nucleus, so that are all the readouts for that your ER stress is high. Your ER stress is high. If they are high, so your these all proteins, these functions will be high. If it is decreased, then everything will be decreased. So this is how they are all intermingly working together. But our hypothesis was there's a protein called CTGF, connective tissue growth factor. Whether it has any role in this ER stress, whether any folding or unfolding of proteins is happening here, for sorry, just thirsty. So whether they are having, because whether if we are having a CTGF up, right, if CTGF is up, then whether ER stress is increased or not. So that's our line of interest is. That's we are looking for. Or if it is decreased, vice versa, ER stress is decreased, or other way around also. If ER is increased, then CTGF is being affected. So that's the whole mechanism to check it here. So to do so, what we can do is we can check protein expression. So whatever things, so first is your DNA, right? First is your DNA. Sorry, my writing is like this. Then is RNA, mRNA, then comes your protein. So if we check everything at protein level, we might be quite sure that things that we are working here, we are working at the right state, in the right manner. There is nothing wrong that we are going through. So mostly what we do as a scientist, we check everything at the protein level via technique called as uh, Western blotting. So there will be one day that we will be doing the Western blotting also in detail. So a Western blotting technique, you can understand the protein expression of a protein. So that was the basic of ER stress signaling pathway. So let's continue. So before we start, this is just the protein structure of CTGF, that it has four different subunits, and they could be cutted out and they could make many disulfide bridges. Disulfide bridges are made between two cysteines. Oops, sorry. They are made between two cysteines within the protein. So it has around 39 cysteines. So they could make disulfide bridges between the different proteins. So these are the various cysteines have been noted out, and they are making disulfide bridges in between them. So if they are broken, they could do the folding. They could make the folding of a protein different. They could unfold and unfold because of these bonds actually. So that's how. So we are very much interested in the disulfide bridges in our case of studies. So we want to check in our so students, this is also, if you have a presentation day, so this is the how you have to represent your things. You have one mechanism page, then you tell the structure of a protein or something that you are looking for. Introduction, this is all introduction is going on. Then you bring up the hypothesis. So we want to test the hypothesis that is CTGF affected by redox or ER stress in cardiomyocytes, or other way around, is the ER stress or UPR affected by the expression of CTGF. So we want to check either way of doing so, and we have told that we will attempt these experiments by using non-reducing gels and incubation with the redox and ER stress eliciting agents. And we also use siRNA technique to knock down CTGF experiment in cardiomyocytes and assess the impact on the various ER stress markers. So in general, you tell your first experiment that what you're looking for, and then you tell how you're gonna look it for. So various methods. So we check. So this is your Western blot figure. So here these are the non-failing. These are non-failing proteins. Then these are ischemic proteins. These are diacardomythy proteins. So these are heart samples actually. We got it from the human sample from the hospital. So we want to check what is expression of our CTGF. So CTGF expression seems the more the darker of the protein expression, the more it is actually. So and this is tubulin is your control. So you want to check one protein that present in each case to be equal, so that we can make a base out of it, use it as a control, divide by the same thing. So CTGF expression fold of control. So this is non-failing ischemic and cardiomyopathy, and this has been done with using software Prism. So we never use Excel or any other way of doing it to make your graph. This is quite professional one for using in the uh, case of your uh, yeah, scientific publications. You will also use it in future, Prism, to make your figures in future. So and furthermore, then you do statistical analysis. So in my case, I have 4 samples. So and I use one way ANOVA test, because this one way ANOVA is done test is done to check within the different groups. And I use Bonifari multiplication comparison test, and to check the significance, the p-value should be less than 0.05 versus a non-failing. So these are the main criteria to see the significance, and we can say that with respect to non-failing and cardiomyopathic uh, heart samples, this protein was quite high. The same way you can analogy with the plants, like so you can have some disease plant. One would be happy plant. Yeah, this is one is disease. This one is happy plant, and so disease and happy. So somehow the protein that we were looking for might be highly expressed in disease plant. It might be low expressed in in protein in this plant. So we can check with the Western blot to start of experiments that whether the protein interest is it high or low. If it is so, then we can increase or decrease it later with the help of genetic engineering by mutation, site-genic mutagenesis, or any other technique that is will be viable for it. Okay. So next, so these are all diagrams, bache, so I will not go through in detail. So my main point was to explain you how a Western blot is done, how you can check things, and how you can find the results out of it by using statistical analysis. And remember, each experiment has to be done 3 times. So all these experiments has been done at least 3 times. So this is your n number. So this has been done from 3 to 5 times. So this is the n number. It has been done n 3 to 5 times, and so because if your n is not more than 3, it will be very hard to analyze the stats of it. At least n should be 3. That is your number of experiments. So same way I did some experiments with oxidative stress with H2O2, then with reducing agents like DDT, and check what happened to CTGF. So there was some increase in CTGF, and we saw some change in the Western blot pattern. Dimer formation was happening. So that means that there's some disulfide bonds were happening. Um, it's you don't have to worry about these data. The main idea, did you understand what was my main idea? Like, you can do plenty of other experiments by adding various other pharmacological agents to it. So I've added thapsigargin in this case, and you do what you do is you do time versus concentration experiments. So here I did a time experiment with 2, 4, and 6 hours of 3 micromolar thapsigargin, and I checked the same thing in the different protein of ER stress in CTGF, in RE1 alpha, uh, and PDI, all the ER stress proteins. So I could see that CTGF with thapsigargin, which is a ER stress inducing agent, so with the ER stress being induced, it was getting increased. So didn't saw with the tunicamycin other pharmacological agents, so that means it was not working out somehow. So here also I use, you can see, I did one experiment as a minute time. This is time time dependent, and this one is concentration dependent. Okay, and so in concentration dependent, oh, actually both are time dependent, sorry. This one is concentration dependent and these two are time dependent, and in these two time dependent, what we can see is that I used a minute first, like till 30 hours of windows is there, 30 minutes of window, and in here it's like 6 hours of window. So like in 2 hours of intervals, 2, 4, 6, and here I use 3, 6, 10, 15, and 30. Like this it was going on. So here is a concentration dependent of DDT. Uh, this is also time dependent experiment, and this is concentration dependent experiment. So here you can see concentration here, and this is here is time dependent. So that's you have to manage what is best for you to see the results. You can do it on your own. So I did these experiments just by seeing that what will be the best way to see the difference in my plots. So I found these to be best while doing multiple experiments and getting to the results then. So I hope these things are clear to you. So this is further heat shock, some more experiments. So this is XBP1 splicing. This is mRNA blot. So what I did is first I check the various pharmacological agents on my samples. What happened to my CTGF protein and other ER stress aging agents. Then I went to the check what if I decrease CTGF in the cells. What if I remove CTGF from the proteins? So what will be effect on the ER stress? So first I check at the mRNA level, then I check at the protein level. So at the mRNA level, I saw that there was a splicing of mRNA by XBP1 was happening very nicely, whereas all other markers were not changed. So at mRNA level, XBP1 was showing things. So that means if XBP1 is being spliced in mRNA, so RE1 alpha is playing role in the ER stress. So ER stress, when CTGF is decreased, so ER stress is increased because we can see the splicing of XBP1 here in the middle one. So that's how everything is correlated and interconnected with each other. So just absorb these things. This will be too much for you maybe to take in, but this might be your daily routine. If you start your research, you will be doing this on the daily basis by knocking in one gene, then checking what happened to other genes, and then going through that further. So that was done at the mRNA level. First you check at the mRNA level, then you check at the protein level. Yeah. First you check at the mRNA level, then you check at the protein level. So at the protein level, most of the RE ES stress proteins like RE1 alpha, BIP, PDIs, they were all decreased significantly. And also there was ATF6 was also in the normal case, control case. If you have seen in quite detailed manner, it was present outside the nucleus. But after the knockdown, it's came to inside the nucleus, all of the ATF6. So this proves that that CTGF is playing something with the ATF6 protein also. So these were microscopic conditions. So here the blue thing is you're seeing a DAPI dye. This is the ATF6 antibody. This is CTGF antibody. So this is showing the knockdown of CTGF is very successful. There's a less expression of CTGF, and the merged version is looked like this way. So that's it. So these were the summary of all the proteins. So at the end you present one conclusion, what is doing what, then you tell the future perspective of it. Then you say thank you to your group, and that's it. That was published. This work was not published, but my previous work of my first thesis was published successfully in the scientific reports in Nature Journal, one of the Nature Journal paper as a first author. So that's the first page of that. So some other day we will discuss about this story that is more into the structure biology, which is might be not interested to you guys. So that's why I kept it away. But I hope my main thing was to teach you these protein data, mRNA data, and Western blotting. How you understand, how you make concentration dependent experiments, how you make time dependent experiments, all these things. So that's it. So please check to the general people if it is paid or not. Does RE1 alpha and CTGF work in the replacement? If like one expression is down, other another will work in the first place. And what is the essence of tubulin protein to compare its span with CTG and Western blot? Because tubulin is a protein is present in most of the in all of the cases. So we used mostly it's universally accepted that if you're doing experiments with CTGF, tubulin will be the most appropriate control because it will be not affected in any case with the other experiments. It will remain constant. So that's why we are using it. Yes, that's what we discovered, that RE1 alpha and CTGF they are interconnected with each other. So CTGF might be with increase in with the decrease in its concentration, we saw decrease in protein expression of RE1 alpha. So they are interdependent with each other. But how? What could be the reason? Whether they are helping in the folding of a protein or maintaining the ER stress homostresses of the protein. So there are many questions to look for. So for that we have to do many other experiments like co-immunoprecipitation experiments to check whether they precipitate together or not. Or we can do DNA chip to check at the DNA level whether they are working at the transcription factor maybe or not. So there are many things, many unsolved questions in during that time, but we just finished that project there. It was a small thing just to see how things were working out. So Let's come to the real point now, of our today's lectures. That is somaclonal variations. Uh, so we are starting with the unit 2. We are done with the practical part yesterday, most of the things. Demonstration of gel electrophorese, isolation of DNA, media components, tissue culture, requirements for plant tissue culture. Some of the methods we also did in between. So that will cover it. So, but we are done with unit 1 also. So today is the unit 2, which is a bit tricky part, but we will go slowly, and let's go how things works out. Um, so so this is the main idea that I have divided this lecture into 11 parts, including introduction, its definition, how you can isolate somoclonal variants. Then there's a screening part is quite important with R0, R1, R2, three different regions you can screen your somoclonal variants. Then it's how to characterize them, then molecular basis of the somoclonal variants, then origin, how it is origin of somoclonal variation. It could be in vivo, novel, or factors. Then factors affecting somoclonal variations. Then there's a gameto clonal variations. Then how somatoclonal variations and induced mutations they are working together. Then some of the achievements from somoclonal variations. What are the advantages and the limitations? So we will be dealing throughout to all these chapters today. Um, so genetic variability present among culture cells. Plants derived from such cells. So there is some genetic variability present in the culture cells, and plant derived from these culture cells or progeny of such plants is called somoclonal variations. Plants having some genetic variability. So plant generated from the culture cells constitute first one are the R0 generation, then the sexual progeny from R0 to make it to the R1 generation. So in case of asexual propagated cells, R1 generation will consist of asexual progeny of R0 plants. So this is like R0 and R1 generations are there, having a genetic variability in the cultured cells. So how can you isolate your somoclonal variants? So very nicely shown to you here. That is, you have your X plant. Then from that X plant, you make a callus or suspension culture. From this callus suspension culture, you regenerate either on the go on to the left side regeneration by having a plantlets out of it. The first generation would be R0. Then you do the sexual progeny of it. Then you have individual progenies known as R1. Then from R1 you evaluate which one that you required and you select that one part. Then after that selection and evolution, the progeny that will be coming out to you, it will be the individual plant progeny of R2 variant. So after from the R1 to R2 variant, then you again do the evolution and selection to check the from the selected plants that are the mutants one that you were looking for, that really that they make sense for you. So this is how the long process you make two times your evolution and selection, and then out of all R0 and 1 and 2, then you have your screening of your plants at the end. Then on the other side, on the right hand side, you can see you have your cell selection. Then variant cell clones are there. Evolution in vitro is done. Then stable clone. Then regeneration is done out of that. Then plantlets are created R0 here. So here we have two steps further. After the cell selection, you have variant cell clones having a variant genetic variability. Then you select in vitro those cells. Then you find the one of the best stable clones out of them. Then after choosing the best stable clone, you go for Sorry. Uh, you go for plantlet, then you again check the evolution of the sexual progeny. So the individual plant would be one. Then you have after R1, you again select your progeny of your mutants. Then the selection is done later on with that. So so that's how the mainly in two different parts you can do your somoclonal variants isolation in this way. So it is observation of large number of cells of regenerated plants for the detection of the variant individual. So individual who has variant out of the cells. Only in that case we do this in observation. So, screening has been profitably and widely employed for the isolation of cell clones that produce higher quantities of certain biochemical. So, this screening has been profitably and widely employed for the isolation of cell clones and has been very high quantities is required certain biochemicals in this case. And this approach is the only feasible technique for the isolation of if the mutants for yield and yield traits. If you are looking for feasible technique for the isolation of yield and yield traits. So in general, R1 progeny are scored for the identification of variance plants. So to find the variant that you're looking for, then R2 progeny lines are evaluated for the confirmation. So it is desirable to avoid screening R0 plant and particularly for yield and yield traits since how traits are greatly affected by the environment. So that is how low variable heritability and R0 plants would be greatly affected by the in vitro culture regimes. So it's stressful processes of the establishment in the soil. So this is just general in telling you in the previous methods that how the evolution and selection is done, the screening is done. So screening at the end is done. They hear this one. So they have explained that at different level R1 and R2 and R0 to check whether you have a good yield and there is no low heritability and in vitro culture that you can establish them in the soil very easily. So you have to screen your plants wisely. Then comes the characterization of the variants. So somoclonal variants isolated through the cell selections are often unstable. They are not so stable. So the frequency of stable variants may range from 8 to 62 percentage, perhaps depending upon the species and the selection agents. So several clones lose their resistance to the selection agents after a period of growth in the absence of selection pressure or repeated screening. Such clones are very unstable variants. So they are not so strong variants as per the required one. So some variant phenotypes are quite stable during the cell culture, but they disappear when plants are regenerated from the variant culture. Such changes are known as epigenetic changes and are attributed to the stable change in the gene expression. So example, growth regulators, growth habituation of cell cultures. So how can you characterize your variants? So first you have your explant, right? Then you evaluate and select like we did in last one. Then you have a variant clone. Then you again from the variant clone you go for evolution and selection. If there is a wild type phenotype that you not required, then you escape out of it, then you remove it. But if you have variant phenotype that you're looking for, you make a variant out of that. Then from that variant you again go for evolution and selection after growth in the absence of the selection agent. Whatever the selection agent that you have added you want to see, then you do it in the absence of that. Then wild type phenotype, if it is unstable, then you escape, so you don't take it. So but with the stable variant, if you have a stable variant further, in the stable variant uh, you go for regeneration of plant. So if there is an epigenetic variant, so something wrong at the DNA level that you were looking for, it's not coming as it should be, then you remove it. Otherwise, if you have stable variant again, who has epigenetically quite stable, then you go for further again. Then you go for sexual and asexual progeny. Again, if you have not good DNA epigenetically variant, then you remove it. Otherwise, you have your mutants at the end, phenotype mutant. So here, like we keep, it's like yes or no, yes or no. Like in the computer science students, they do yes or no. So if it is yes, what you will do. If it is no, then what you will do. If it is yes, then you go like this. If it is no, then you don't go like this. So that's what mainly happening in these variants so far. So if things are coming out for you, then you go for it. If it is not, then you don't go for it. So this flow diagram depicting the characterization of the cell clone obtained through the screening selection method. So some of the molecular bases of somaclonal variants, how could you select them? So there could be change in chromosome number at the end. There could be, oops, there could be change in the chromosome number. Gene mutation could be changed. Plasma gene could be changed. Alternation in the gene expression, gene amplification, mitotic crossing over, transposable element activation, rearrangement in the cytoplasmic genes. So all these things could happen in our at the molecular basis with these somoclonal variants, because they are genetically we are checking everything at the gene level. So if we are checking things at the genetic level, so we could occur some problem also at the genetic level also. So most mutant isolated from cell cultures may involve single gene mutation. So here we want mainly a single gene mutation, and the mutant element being either dominant or mostly recessive. They will be either dominant or mostly recessive genes. So gene amplification. This is increase in number of copies of a gene per genome of the organisms in comparison to the naturally present. So has been reported in some variants, usually recovered through the stepwise selection of plant cells in vitro. Okay, so there could be de-amplifications, may also occur in the somoclonal variants. Example, ribosomal RNA genes could be also be used in that case. Then, as we discussed, sorry, mitotic crossing over. Oops. This could be also account for some of the genetic variations detected in the regenerated plants, leading to the recovery of homozygous recessive single gene mutation in R0. At the R0 generation, you could see some changes, single gene mutations because of the mitotic crossing over. So there could be change in the chromosome number and structure of your plant, cultured plants, and these changes could be recorded in and regenerated in the cultured cells later also. So in many cases, there's an alteration in the organellar genomes have been reported. So like mitochondrial DNA being more frequently affected than the cytoplasmic DNA. So your mitochondrial DNA is being more affected during these changes of the somoclonal variant. So be careful. We have to be just take care of all these things when we are working with them. Then origin of somoclonal variants. These are recovered maybe in two different contexts, arise from the in vivo process or from the in vitro culture. So 90% of angiosperms differentiation is accompanied by endoploidy or aneuploidy, uh, which is known to occur in vivo, whereas somatic embryos were regenerated directly from the explants without an intervening callus phase. On the other hand, frequency of somoclonal variations increases with duration of in vitro culture. So in vivo phenomena scaled up in vitro. So some of the things to study further in this case is that to check the somoclonal variants in vivo that may simply magnified in vitro. That is endoduplication, mitotic irregularities, errors by DNA polymerase, and base changes in DNA. So all these being dealt in this case. So endo first we will deal with the endo reduplication. Mainly what happen is during your mitotic phase, when your cells are dividing at the meta phase, so there is a cytokinesis is happening that the two different cells are being made. But in this case, that phase is not possible anymore. But you have two more. It's not happening, but you still have some cells might be dividing, some cells might be not dividing. So you will have cluster of cells also with different DNA, but also you will have one big cytoplasm with one big nucleus where inside many cells are dividing. So that's known as endo reduplication. So it is divided into different phases. As we know, this mitotic phase, that is G1, S, G2 and M phase. And in higher plants, cells that complete S phase uh, invariably enters M phase, and cells that become quiescent. So after completing the S stage, most of the plants they enters the M phase where they become stop dividing and enters the G0 phase, which is equivalent to G1 phase. So cells that has completed S phase can enter S phase only after it passes through the M phase. So strict regulation is due to protein complex called lysing factor. So here a lysing factor is there, which is produced in the cytoplasm and is unable to pass through the nuclear envelope. It does not pass the nuclear envelope; it's remain in the nuclear envelope, and because of this, there is a strict regulation of protein mitotic phase is happening. So 90% of angiosperms, proportion of differentiating cells, do not only enter M phase after completing the S phase, they may also enter the S phase or two or more times, one after the other, without an intervening the M phase. So results are as follows. You can see that it's producing DNA content in the differentiated cell forms with 4C and 8C, and this phenomena is called endoreduplication. So you have your S phase. As you know, the licensing factor could not go outside, so it's remain inside. But in the M phase, it tries to go outside. So licensing factor ensures that the cell does not enter the S phase. And again, before going through the M phase, before it goes to the M phase, it just confirmed that it should not go to the S phase. So it's essential for the onset of the S phase. So it's in LF. Where is your LF star is the inactive licensing factor. So LF1 is the active part. So to explain it further, there is one small video. So have a look.
There are three separate mechanisms through which a cell may possess more chromosomal material or more nuclei than the typical mononucleate diploid cell. Cells may replicate their DNA but fail to undergo mitosis in a process known as endoreduplication, so that a single nucleus has multiple copies of chromosomes. Cells may undergo mitosis and replicate their nucleus but fail to undergo cytokinesis, and so that a cell may have separate nuclei. And then finally, a single cell may form through the fusion of multiple cells. All three of these examples are known in the human body.
So, that was something to explain you with the figure, with the animation. Um, So, besides that endo reduplication, there could be mitotic irregularities or errors by DNA polymerase. In the mitotic irregularities, it could be done because of chromosome breakage, anaphase bridge, lagging chromosome, etc. could be the reason behind it, leading to the low frequency in the root tips. You will have less division of mitotic cell division, and also the PMS, the polar mother cells, meiotic cell divisions. We will have low frequency of them. It could also be due to aneuploidy and structural chromosomal aberrations. Could also lead to this phase of it. So there could be errors by DNA polymerase. So DNA polymerase commit errors during the DNA replication, so which incorporates wrong bases in the newly synthesized DNA at frequency of 10 is to power minus 5. And bulk of these errors are corrected during the proofreading by this enzyme. So since plants cultured in vitro divide repeatedly, these errors will accumulate and increase the duration of the in vitro culture. So it's something about how DNA polymerase could play a role by changing the synthesizing the wrong base pairs with at the frequency of 10 raised to power minus 5. So there could be bases could be changed in the DNA. Major cause of spontaneous mutation in E. coli is being deamination following modification of the DNA bases. So small proportion of C residues is methylated. They are methylated to yield 5-methyl cytosine, and an appreciable proportion of 5-methyl cytosine undergoes spontaneous deaminations to yield thymine residues. So this produce a GC80 transition in case of residues are known to be methylated in CpG and CpX-PVG sequences. So that's how there's a transition in itself of your uh, base pairs. Base pairs are being connected to a different part of it. So it changes many things. Further, uh, to take you now, we are done with the somatoclonal variants. So there is also called thing termed as gametoclonal variants. So this is the main question being asked mainly in these terms. What is the difference between somatoclonal and gametoclonal? So this is gametoclonal is basically dealing with the genetic variation present among the culture cells derived from the gametophytes or among the plant regenerated from them. So such cells are obtained through anther or pollen culture haploid. Main features are as follows. First is selection should be preferably done at R1 generation onward in order to avoid any confusion effects of tissue culture. There should be no confusion or anything, and you have to done it in a right way to select your R1 generation. Then comes the utilization of gametoclonal variations can be only possible after there is a chromosome number is doubled, and chromosome number is doubled with the help of colchicine. So with the help of colchicine, you increase your chromosome number, and you can see the changes at the at the gamete level. So further colchicine treatment may generate additional genetic variations. This effect is largely genotypically dependent. So these colchicine treatment may generate additional genetic variations. So this is all in all is dealing with the genotypic independent. So this is how it is shown. One of the example here we are showing you commercially development of acceptable line of tobacco from an inferior plant, whereas potato virus strain Y33 31 PVY potato virus PVY resistance gametoclonal variants by backcrossing to the parent variety followed by selfing and selection. So you can see here is this is your parent plant McNair 944. So with the help of anther culture you produce the haploid plants first of all 545. Then you added your strain, your potato virus strain. So out of all your plants one plant survived. Then you did the chromosome doubling with the help of colchicine, uh, and also you add PVY strain, the strain also, and you did the yield test. So you have afterward all these experiments, you have your first generation as a PVY resistant genes with a 19% lower yield. But what you do is with this lower yield, but you it's a virus resistance, you backcross with the Mac9 with the parent plant, you do the selfing and selection, then you have a uh, so you have a yield which is comparable to the McNair 44. Your yield will be also equivalent to that the parent plant and likely to be released for commercial cultivation. So it's ready to go for commercial cultivation also. So that's how the gametoclonal cultivation is mainly done. So somatoclonal variation induced mutations are preferably to induce mutations for following reasons. Chimerisms not occurring. Sometimes sterility is there to avoid somoclonal variations. New alleles and even new mutations could be recovered from that. Frequency of a useful mutation is very high. Highly effective selection can be applied in vitro for several economic factors. Large number of individuals can be secreted in vitro. So these are the following regions to induce mutations in somaclonal variants. So these are the frequency of some somaclonal variants being produced, screened, and their resistance percentage out of that screen, that they are comparable to the control. And so there you can see sugarcane, potato, and apple. And the sugarcane is fight against the Fiji disease. So somaclonals were screened around 235. So resistance somaclonal percentage was 23 around. So comparable to the control. So there was a 5 percentage. They were more resistant than the control. This then there is a disease early blight we know, and a late blight by Alternalia solani and Phytophthora infestans. So here we have somaclonal screen 800, and out of them resistance to the race was 2.5 percentage, and resistance to race 1, 2, and 3, 4 0.5 percentage. So they were 100% in scab disease. They were 100% clones, subsoma clones were screened resistance to the disease. So more than, and they were more than resistant than the parent variety. So that was quite high advantage of these mutations to the plants. So, how do you cell selection has to be done in this case? Cell selection strategies have been devised for the isolation of variety of useful variants, such as for disease resistance, for abiotic stress resistance, for efficient nutrient utilizations. So, plant material used for cell selection in this case. While you doing your create your variants can be applied to the X plants, callus culture, blighted plants, suspension culture, protoplast, or then generally callus species are used in the selection experiments. Cooperative cell death has been taken care in the case of wild type animals, leading to the increase in frequency of unstable variants. Suspension culture permits a more uniform exposure of cells to the selection agents, and use of platelet cells or protoplasts minimize many of the problems of encountered with the callus and suspension culture. So use of mutagens treatment of cell culture increase the frequency of variants, and sometimes did not have significant beneficial effects. On the other hand, it could also give a deleterious effects like increasing the sensitivity of the callus to the toxin, mutagen-induced bleaching, and necrosis. So selection for disease resistance variants could be pathogen, toxin, pactolytic enzyme, culture filtrate. All these things could be done for the selection of disease resistance variants. So what are the achievements that we have seen so far? So you can see that alfalfa, crop, Brassica junicea, carrot, all these plants have we have identified somaclonal variety of them. These were the parent variety, and we have increased their yield was increased. We have increased their yield resistance against the diseases. We have low neurotoxin and also there are some with a salt tolerant, immune to rust. So there are a lot of varieties have been produced with the help of somaclonal variants in various species. So 60 varieties has been done so far. And somaclonal isolated have 20 different crops like asparagus, sorghum, wheat, maize, rice, tobacco. There has been a coffee improvement has been done also. More than that, scarlet, sweet potato cultivars, shoot tip cultures have been derived and clones yield and resistance. And oh no, the Fiji disease resistant sugarcane variety has yield advantage of it. So these are the various advantages of the achievements that we have seen. So what are the advantages that we can see from the somaclonal variants occur in the in the case of species is that they occur in high frequencies, and they are quite advantages over the conventional mutagenesis techniques that we do. So because we do a lot of selection, we do a lot of screening, we do a lot of uh, yeah, a lot of check is there, and lot of crosses are there with the self. We also check the yield is also good, high is also high, and they are also resistance to the disease. They are also resistance to the salt tolerance. So whatever the things that we are looking for, so we keep on track on our things. Beyond that, it's a new allele or new mutation may be isolated, which were not available in the germplasm or through mutagenesis. So, example is a jointless pedicle mutant in tomato. So this was one mutations done in jointless pedicle mutate in tomato was done. Time decreased to 2 years for the release of new variety. Earlier it take us to 5 to 6 years, but now it takes very less years to do so. From so undesirable features should be will be not there, like sterility, that can be taken care of. Further, effective selection can be practiced at the cell level for several traits. Example: disease resistance, small effort, time, cost, space requirement. Very less is required in this case. Um, Simple and cheap for plant biotechnology compared to the somatic hybridization and genetic transformation. All those techniques that they're there, so it is quite simple and quite better in plant biotechnology level of technology so far. And in vitro regenerations of tomato and potato shows increased rates of combination, so we saw a lot of combinations in them also. So what are the limitations? Uh, that we have cell lines reduced or no regeneration potential in them, or the clones that we are producing, they have reduced fertility, growth, unstable, and even some of the performances are even overall not so good. So sometimes there are also backlags for the this technique. Agronomically, traits like yield, quality are not expressed at the cell level, not selected for in vitro. If they are not happening at the cellular level, then we do not go for further in vitro experiments. Pleiotropic effects can be done by transferring the mutant genes in the different genetic backgrounds selected for the desired combination of traits. Beyond that, variations may be not novel but may be useful. So, depression in the quantitative traits of the most of the variants are one of the key things are happening in this case. So thank you very much, class. So that was something about the world of somaclonal variants and gametoclonal variants also, how they are working, how what was their molecular basis of inheritance, their process, their advantages, their limitations, their drawbacks, everything we have colored quite in a nutshell. So we are done with this, so we will take so this we will not go. And guys, would you like to go for the first part of thesis also, or that was enough for you? Because that will be something about structure biology. If somebody is interested to study about structure biology, we can make that later part. Just let me know in the comments. Would you like it or no? So I will take some 10 minutes break now and then see you at 21:10 then. Take a break. See us as a fresh look then. Okay? Wakey wakey, awake everyone. Awesome. See you then. Take care. So, welcome back, class. Let's continue the next topic. Somatic embryogenesis and synthetic seeds. So, this is your second topic from your theory, from your all over things we have done. This will be dealing with the somatic embryogenesis and synthetic seeds. So we are done that which is not your zygotes but something somatic embryos are formed. That is mainly your somatic embryogenesis is. And with the help of somatic embryos you can create synthetic seeds by encapsulating them with the water gels or some hybrid gels out of them, and you can use it for further future for your experiments. So this section has been divided into 10 parts of your somatic embryogenesis and synthetic seeds. That is, I will be dealing here with the introduction. What is in vitro somatic embryogenesis? Organogenesis versus embryogenesis, how they are being differentiated. Factors which are affecting the somatic embryogenesis, embryo maturations and germinations will be also dealt with this topic. Secondary somatic embryogenesis, synchronization of embryo development, production of synthetic seeds or artificial seeds, application of somatic embryogenesis, limitation of the somatic embryogenesis. So all these parts will be dealt during this topic. So first is that is somatic embryogenesis is something like embryo-like structure, which is analog to your zygotic embryo, are formed either directly from the tissue or via an intervening callus phase. So this phase, the process is opposite of the zygotic or sexual embryogenesis, and the fertilization process prompt the egg cells to divide and develop into an embryo in the case of embryogenesis. But however, fertilization is not always essential to do so in the case of embryogenesis. Sometimes parthenogenesis could happen and lead to the growth regulators and lead to the embryonic development. So any cells with the female gametophyte, even that of sporophytic tissues around the embryo sac, may give rise to embryo. So, for example, development of adventitious embryo from the nucellar cells could be also possible, which is common feature in the citrus and mangifera crop. However, the nucellar embryo attain maturity only if they are pushed into the embryo sac at an early stage of development, or else they will fail to mature at the end. So this is the structure of your ovule, longitudinal structure. So first is your chalza. Yeah. Then comes your outer integuments, antipodals here. Your three antipodals. Yeah. Then in this will be your inner integuments. Then you have polar nuclei inside of this. This whole part is will be called as nucellus. And the bottom with three things that you're seeing is embryo sac here. This embryo sac, then two synergids, and this whole thing together. So this whole is embryo sac, and this first one is your egg cell, and these are two synergids, and then this is your micropolar region, this one, and this is your funiculus, like ranging to your stigma then further. So that's how each part being described here of your ovule. Okay. Um, Further, in vitro somatic embryogenesis They could be of two type. One could be indirect and direct. In indirect, where it has first the callousing is induced in the explants by rapid cell division. Later, the callus give rise to somatic embryogenesis. Then direct somatic embryogenesis, where the somatic embryos are developed directly from the explant without an intermediate callus phase. So these could be also possible by direct or indirect. So here at the bottom you can see a development of somatic embryogenesis via callousing via direct method. So here first uh, is the globular phase here, this one. Then is your heart shape like this. Then comes the dicot embryo. Then comes your uh, torpedo. This one is a torpedo. So all different shapes are being formed, and hence at the end your callus is made from them. This one is the direct somatic embryogenesis, showing you where embryos with a various stage of developments. So how it is done nicely. So in the area of in vitro somatic embryogenesis, you have following stages. You can see there globular, hard, torpedo, and cotyledon stages. So these are the complete stages that is happening in the zygotic embryos, which resembles like so somatic embryo resembles the same like zygotic embryos in their case. So embryos germinate and develop into complete plantlet. The only major difference between somatic and zygotic embryogenesis is the somatic embryos do not pass through the desiccation or dormancy phases as it happens in the zygotic embryos, but rather to continue in the germination phase. So here at the bottom, you can see there the first is the globular stage, then comes the early heart shape, then the late heart shape, then the torpedo shape, then early dicot, then fully developed dicot embryo. So these are the zygotic embryos, but somatic embryos are also the same like stage, but in that case, desiccation and dormancy phase is not happening. That's the main difference between these two. So in vitro somatic embryogenesis, whether originating directly or indirectly via callousing, somatic embryos arise from the single special cells within the cluster of meristematic cells. So in callus, mass, or in the explant tissue, somatic embryogenesis is regarding the three steps. That is induction of your embryo, embryo development, and embryo maturation. So all these parts are being taken care while doing your experiments. So first is induction of embryo, then embryo development, then embryo maturation. Then difference, what is organogenesis versus embryogenesis? In tissue culture, plant generation by somatic embryogenesis may offer many advantages over organogenesis, such as embryo is bipolar in nature than the monopolar in organogenesis. So embryo arises from the single cell and have no vascular connection with the maternal callus tissue or the explant, cultured explant. On the other hand, during organogenesis, shoots or roots develop from the group of cells, resulting into the chimera formation, which later establish into strong connection with the maternal tissue. Further, somatic embryogenesis requires a single hormonal signal to induce bipolar structure capable of forming a complete plant, while in organogenesis it requires two different hormonal signals to induce shoot first, then root organ. So that's the main difference between your somatic embryogenesis, in which you just need single hormone to grow, where to grow into to do a bipolar structure to leading to the complete plant. While in organogenesis, you need two different hormones to induce shoot and then root organ. That is your oxygen to cytokinin ratio. So you have to maintain different cases in that case. So genotype factors affecting somatic embryogenesis. So it's first is genotype and type of explant. So these are the first, like in organogenesis, somatic embryogenesis is a genotypic dependent, and for a given species or variations in a response between cultivars have been observed. Several plants like wheat, barley, soybean, rice, alfalfa, they're all dealt in with this somatic embryogenesis. And genotypic variations uh, could be due to endogenous levels of hormones. Therefore, if a species has not seen a somatic embryogenesis previously, it required test number of different cultivars. And the explant selection is much more important than the media selection here for somatic embryogenesis process, because immature zygotic embryos have been proved to be best explant to raise embryogenic cultures as somatic embryos, and will form more readily from cells that are already in the embryonic state. So factors affecting somatic embryogenesis, that is in Azad Rishta indica, that is your neem plant, Azad Rashta indica. So there is a immature zygotic embryo at different stages of development. That is globular, early to late heart shape, torpedo, early dicotyledon stage. So we can see all these stages in them, and when culture showed varied potential for somatic embryogenesis. So globular embryos did not show any responses, whereas the older embryos germinated formed calli or different three types of organized structure. That is shoot, somatic embryos, and neomorphs. Neomorphs are the abnormal embryo-like structure with varied morphology. Further factors that are affecting are the growth regulators. So auxins play a major role in them in the somatic embryogenic system, such as carrot, coffee. Most of the cereal require synthetic auxin for the induction of your somatic embryogenesis. So followed by transfer to an auxin-free medium for embryo differentiation. So synthetic auxin like 2,4-D is the most commonly used. So sometimes we also use NAA, IBA, picloram, IEA also been used. So IEA is a naturally occurring Indian indole acid is a naturally occurring auxin, but is a weak auxin, more readily broken down compared to the 2,4-D and NAA. But we are mostly using synthetic auxin by used is a 2,4-D in our case. Then in the growth regulators in continuation, a natural occurring auxin like this we did. So the auxin, particularly 2,4-D, if we put them in the concentration of 0.5 to 1 milligram per liter, it will stimulate formation of localized group called as PAMs. PAM is pro-embryonic masses. So this is our embryogenic cells with PAM indicating the arrows, and these are cluster of cell populations component to the somatic embryos. So they are quite good to go for your somatic embryos in the future part. So in repeated subcultures on the proliferation medium, embryogenic cells continue to multiply without appearance of embryos. However, if the PM are transferred to medium with a low level of oxygen or no oxygen in the medium, they develop into the embryo. On the other hand, the presence of oxygen in the proliferation medium seems essential for the tissue to develop embryos in the ED medium. So they are also quite essential for the tissue culture also in your ED medium, that is your embryonic development medium. So we have discussed a lot about auxin here. Now comes the cytokinin. So cytokinin in general directly induce the somatic embryogenesis with coalescing of explants. In most cases, TDZ is a cytokinin which is herbicide which mimics both cytokinin and auxins, and they are being used when zygotic embryo are quite essential for as a explant source. So most commonly used cytokinins are BAP and zetin. So in the case of cytokinin, we use BAP and zetin, mainly used to extract your zygotic embryo for your explant source. So this is showing you an explant showing differentiation of neomorphs and somatic embryos. This is neomorphs here on this side, and these are the somatic embryos on the same explant. So in this neem plant, what we are seeing is somatic embryo differentiation was induced by the culture of mediums. So somatic embryogenesis at maximum level could be seen with the help of barb containing media when early dicotyledon stage of embryos were cultured. So medium with the 240 induced only neomorphs. 240 will induce neomorphs directly from the explant. So every each medium, each thing has its own use, its own level of work, its own level of working out there. So factors affecting somatic embryogenesis further to take care. So these are the figures showing you in the A part. An explant is showing you different direct differentiation of neomorph. So your neomorph is being differentiated. Some of these showing like structure like cotyledon like flaps. So they look like flaps, cotyledon like flaps, and they are in contact with the medium and has been proliferated into the brownish green colors. So it's brownish also, green colors also. And in the B section, you are seeing the histological section of the same. So showing you epidermal origin of the neomorphs of the various shapes, well differentiated, nicely done. Of the explant showing paravascular strands also. Okay. So here, factors affecting somatic embryogenesis. What are the factors that are affecting somatic embryogenesis? That are also third one is now is nitrogen source. Nitrogen source is also affecting it. The most important nutrient of the culture medium is nitrogen, which affects somatic embryogenesis significantly. The form of nitrogen have been strong influence in the induction of somatic embryogenesis. Often the ammonia that is playing the source of reduced nitrogen is required in the form of glycine, glutamate and casein hydrolysate. In few cases, with the medium with potassium nitrate as a source of nitrogen fail to form embryos upon removal of oxygen. However, addition of small amount of nitrogen in the form of NH4Cl in the presence of 55 millimolar of KNO3 followed embryo development. So by changing these chemicals, by changing uh, their volume, we can initiate our somatic embryogenesis also. So nitrogen is playing big role. Our growth regulators are playing big role. Our genotypic explants that you're taking, that is playing big role in the somatic embryogenesis. And then about secondary somatic embryogenesis. In this, mainly your new somatic embryos are proliferated from the originally formed primary somatic embryos, and secondary somatic embryos is reported to have some advantage over the primary somatic embryogenesis, such as they have high multiplication rate, long-term repeatability, and independence of an explant. And by the repeated secondary somatic embryogenesis selected embryogenic lines, we can maintain long period of them in large quantities until the lines have been tested in field conditions, particularly in the prenatal plants. So, for example, in the Azarita indica, the neem, for secondary somatic embryos, primary embryos were used as explant when cultured on the medium with the TDZ and GA3 growth hormones, and further secondary embryos were differentiated directly from the hypocotyle regions without any intervening callus. So, whereas a combination of BARP and indoloacetic acid resulted in the secondary acid preceded by callousing of the primary somatic embryo. So, secondary somatic embryogenesis. So primary somatic embryo showing a direct secondary somatic embryogenesis. Indirectly somatic, indirect secondary somatic embryogenesis. So this is how we are showing you primary and secondary, direct and indirect. So this is about now synchronization, how they synchronize your embryo development. So generally the differentiation of somatic embryos is done in the semi-solid medium or in the liquid medium with a highly asynchronic medium, which gives a very adverse effect on the germination rate. So synchronization of embryo development is very important for artificial seed technology. Physical separation of embryogenic stages, use of growth regulators to physiological synchronize the development. The fractionation of embryos of different stages by filtration of suspension through meshes of different size of gradient centrifugation. So abscisic acid at low concentration is the most satisfactory substance for this purpose. So for example, in carrot, it inhibits the growth of roots and enhances the suspension with torpedo-shaped embryos. So from these production of from these somatic embryos, you can create synthetic seeds on a very large population. You can use them for the commercial use. So this could be achieved with the help of encapsulating the viable somatic embryos with a protective covering to them. And the coating material could be of several qualities. They should be non-damaging to the embryo. They should be mild to enough to protect the embryo, allow germination, but it must be sufficiently durable for rough handling during the manufacture, storage and transportation and planting. The coat must contain nutrients, growth regulators and other components necessary for germination. Furthermore, the quality of somatic embryo should be good enough that they are all uniform stage with reversible arrested growth and with high rate of conversion to plantlets. So two types of synthetic seeds that are produced, that is desiccated synthetic seeds and hydrated synthetic seeds. So to do so, to production for the desiccated synthetic seeds, desiccated means mainly you're removing the water out of your seeds. So it involves the encapsulation of somatic embryos followed by their desiccation and can be prepared by following methodology. That is, so first you mix your embryo suspension and polyox, this polyoxyethylene. You mix your embryo suspension with polyox, and then suspension was dispensed on a Teflon sheet. Dried suspension sticks to glass plate, and then dried to wafers in a laminar flow hood for about 5 hours until the wafers get separated from the Teflon plate. So polyox is readily soluble in water, dries to thin film. It does not support the growth of microorganisms and is non-toxic to the embryos. Then in the hydrated case, what you do is you create a calcium alginates by mixing your somatic embryos with in first case with sodium alginate. Then drop the mixture using a pipette with 100 millimolar solution of calcium nitrate. Then this calcium nitrate, there will be an ion exchange reaction will happen, and sodium ions are replaced by calcium ions, forming a calcium alginate beads, right? So this is how you create your hydrated synthetic seeds. So following are the applications of somatic embryogenesis, which are as follows. That is, following features of somatic embryos promoted many scientists to achieve regeneration via synthetic embryogenesis using various explants, most popular ones being zygotic embryos or excised cotyledons or hypocotyls. So first case is these SEs offers immense potential to speed up the clonal propagation of plants being bipolar in nature. So as they are bipolar in nature, they could lead to the quite high volume of your potentially you could grow your plants. So being single cell in origin, there is possibility to automate large scale production of the embryos in bioreactors and their field planting as synthetic seeds. The bipolar nature of embryos allow their direct development into the complete plantlet without the need of rooting stage as required for plant regeneration via organogenesis. Furthermore, apodermal single cell origin of embryos favor the use of this plant for plant transformation. It can also be used for production of metabolites in species where embryos are the reservoirs of the important biochemical compounds. The production of artificial seeds using somatic embryos is an obvious choice for efficient transport and storage. The embryo culture technique is applied to overcome embryo abortion, seed dormancy, and cell sterility in the plants. So what are the limitations? If you have seek the advantages, there should be some limitations also, that are um, complete conversion into plantlets or poor germination of embryo is a major limitation in this case. Compared to the other plants, active research is going on to involve forestries have been very slow in this case. Low frequency of true type embryogenic efficiency is also responsible for the reduced commercial applications. And furthermore, last not the least, to obtain a complete conversion to plantlet, it is necessary to provide optimum nutritive and environmental conditions in this case. So thank you very much for today being there with us in a nice audience. So that's the basic two parts we have to do today. They were quite complicated though, but this is quite, yeah, this is very basic things that you have to go through, and it's I have read through the GATE exam also, the GATE syllabus. It's coming in there also, in their syllabus also, in the book of GATES also. So please go through all these terms before you go for further reading. Read them once more because this lectures that I have created like day and night for you, and there's more you read, the more you will understand like how things are happening. Because I can teach you all this, I have taught you everything, but it's now your work to go through it. The more you put your effort to them, the more you will be understanding, getting the grasp of this field. So this plant biotechnology, I could also have teach you other interesting stuff, but I really want that the syllabus by IIR, by Indian Agriculture Institute, Indian Agriculture, ARI. From them, what they are telling us, it is universally accepted, and I really want that course to be completed as per their guidelines, as per their lectures, as per their PowerPoint presentations. So these are all from guided by them. So please go through them once and read them while understanding each terms again and again. So which factor is doing what, how, yeah, various molecular biological methods, all the things that we have done today, just go through them. That's I would like to say at the end of our lecture. So any questions further? So this would be our kind of doubt session. If you have any doubt further beyond studies or any other questions, you can ask me now. So, and guys, please subscribe to the channel. Uh, so after this, if we are still in touch with each other, I will try to make some YouTube videos. So book, I'm preferring for myself HS Chawla. So I can look for if there is any PDF version available. Introduction, plant technology. You can get it at any bookshop there. So this is, I found this book to be quite impressive because all everything has been described in a very simple language and easy manner, so that you don't have to go through very complicated things. And my lectures will be also sufficient for you, as in, sorry, um, because it's included everything in it. So these lectures will be available to you also later. So I will share now with you today's two lectures that we've been through. I will look for some PDFs for related to plant biotechnology. If I could find out, I will share with you all. Yes, yes, I will share. Don't worry. I have a couple of books with me um, for biochemistry subjects. If you want, I can share with you later. Harper, I have. Lippincott, I have. Lehinger, I have. If anybody needs them, please just let me know. Anybody needs these biochemistry books? Because tomorrow there will be one workshop of biochemistry also. Okay. Uh, okay. Okay, I will share Lehinger just now with you. Or just share everything. Don't worry. Okay, done. So any other thing? Um, Give me some time, Surbi. At least you go through my lectures for sure. They will be enough. Give me some time. I will get back to you that these plant biotechnology subjects that we are going through, will it be enough or not? Maybe tomorrow I can answer you. Don't worry, don't worry. We will go through this. Okay, if you have not any further question, I will close this panel today. Thank you everyone for being there, and wish you all a good sleep, a good night. Take care, stay safe. As the corona is started to, the second wave is started to come back again. So stay at home as much as you can. Try not to go outside. Just go for the essential things, and just study at home if possible. But yes, just Chawla I have to look for. If I could find, I will share you. But I have a hard copy at my place. But if you go to any shop and you ask for second hand copy, you can easily get it. It's one of the common one. From the plant biotech books, I think I don't have any. Maybe I can check in my pen drive. During my German time, I have a lot of books at that time. but but don't read too many books, please. That's my request to you. Read just one or two books as a major thing to go for your studies, because too much books makes you confuse for your final studies, so please take care of that. Okay, then good night, take care, bye bye.